Neet biology, diagram based technical terms, 4 to 5. They are discussing the topic about plant growth and development. Important terms. Number 1, abscission. Abscission means shedding of leaf and fruits from the plant. The process of shedding of leaf, fruit or flower by a plant is called abscission. The shedding of a plant pot takes place by the formation of a special layer of cell called abscission layer within the region of attachment. The middle lamella between certain cell in this layer is often digested by polysaccharide hydrolyzing enzyme such as cellulase and pectinases. Certain other degenerative changes also occur, making the region soft and weak. The organ from a plant is easily detached whenever there is heavy rainfall or wind, etc. The abscission occurs due to the change in the hormonal balance. It has been observed that the abscission layer formation occurs rapidly when the auxin gradient becomes less, that is less auxin on distal side than the proximal side of the leaf, flower or fruit. The plant hormone like ethylene and abscisic acid promote the abscission. A high concentration of auxin prevents the formation of abscission layer. Next one, apical dominance. The inhibition of the growth of lateral bud in the presence of apical bud. Apical dominance is the phenomenon by which presence of apical bud does not allow the nearby lateral bud to grow. Lateral bud start developing into branches when the apical bud is removed. However, if a paste of auxin is applied on the decapitated shoot, the growth of lateral bud remain inhibited in the apical bud is present. A plant with strong apical dominance has little or no branching. Example, sunflower. Weak apical dominance leads to bushy habit. Example, tomato. The burning of hedge plant is done to remove apical bud so that lateral bud may be relieved for their suppression. It results in the dense growth of hedges. In tobacco, removal of apical bud is called topping, promote vigorous growth of lateral bud which produce larger leaves. Next one, antagonistic. Effect of one substance opposite to the other activity. The effect of two substances in such a way that they have opposite effect on the same process. One accelerate and other inhibit. Example, abscisic acid and gibberellin during seed or bud dormancy. Abscisic acid induces dormancy and gibberellin break it. Next one, bolting. Sudden elongation of a rosette or condensed stem before flowering. Gibberellin induced stem elongation in rosette plants. Example, cabbage, canpani, etc. Such plants show retarded internodal growth and profuse leaf development. In this plant, just prior to the reproductive phase, the internode elongate enormously, causing marked increase in stem height. This is called bolting. Rosette plants require either long days or cold night for bolting and initiation of flowering. If a cabbage head is kept under warm night, it retains its rosette habit. In cabbage, bolting can be induced artificially by the application of gibberellin under the condition that would normally maintain the rosette form. A weekly dose of 0.1 mg gibberellic acid make cabbage plant to show enormous elongation of stem and flowering. Another one, determinate meristem. The meristem which is active only for a short period. However, the meristem such as those involved in leaf and flower formation are active only for a short period and are primarily responsible for the formation of a organ. These are called determinate meristem. Next one, de-differentiation. De-differentiation means refers to the regaining of the capacity to divide by the differentiated cell. In plants, living differentiated cell can regain the capacity to divide mitotically under certain condition. The sum of event that below this capacity is a differentiated cell to divide once again is termed as de-differentiation. A de-differentiated tissue can act as a meristem. Example, intervesicular vascular cambium, car cambium, and wound meristem, that is called, all derived from permanent cell. Next one, development, refers to the sequence of events that occur in the life history of a cell, organ of organism involving growth, differentiation, maturation, and senescence. Beside growth and differentiation, a plant also undergo development. Development is the sequence of events that occur in the life history of a cell, organ, or organism, which includes growth, differentiation, maturation, and senescence. Thus, the development includes all changes that a plant goes through during its life cycle, from germination of the seed through growth, maturation, flowering, seed formation, and senescence. The term development is also applied to the process of formation of cell, tissue, or organ. It can also be applied for the changes in phases of life cycle of an organism. Example, from vegetative to flowering, from leaf initiation to leaf expansion. The appearance of chloroplast in cell exposed to light is an example of development at a subcellular level. The last phase of the development process in plant is senescence, during which events leading to aging and death take place. The sequence of process which constitutes the development of a higher plant. Another one, differentiation, the qualitative change in the terms of structure and function of newly formed cell. Growth is invariably associated with differentiation. The cell which are derived from the RAM, that is root apical meristem, second one SAM, shoot apical meristem, and other meristem differentiated to and mature to perform specific function. This act leading to the maturation is termed as differentiation. The cells undergo a few major structural changes both in their cell wall and protoplasm. Thus, differentiation can be defined as the qualitative change in terms of structure and function of cell. Although all the individual cells in a plant have the same genetic information and influenced by the same external factor, they come to perform different function depending upon their location inside the plant. The internal cellular mechanism block the expression of certain genes and allow other to show in their effect. Next one, indeterminate meristem, the meristem involved in open form of growth. Plant growth is generally indeterminate. In plant, growth occurs by cell division and cell enlargement. Plant retain the capacity for unlimited growth throughout their life. This ability is due to the presence of meristem at a certain location in their body. The cells of the such meristem have the capacity to divide and self-perpetuate. The product or the new cell produced by the division of meristematic cell, however, soon loses the capacity to divide. The cell which have the losses the capacity to divide and ended into G0 phase of the cell cycle make up the plant body. This type of growth, wherein a cell always added to the plant activity of meristem, is called open form of growth. There is a meristem of the FX, every root and shoot called root apical meristem and shoot apical meristem. The meristem such as RAM, root apical meristem, SAM, shoot apical meristem, vascular cambium, car cambium, and intercalary meristem, which are involved in open form of growth, are called indeterminate meristem. Another one, phototropism, tropic movement induced by direction of light. The tropic movement taking place by a response to light stimulus is called phototropism. Some of the plant parts such as stem, branch, leaves, and pedicel of flower move towards the stimulus of light and said to be positively phototropic, while others such root and rhizoid which move away from the stimulus of the light are called negatively phototropic. Next one, photoperiodism, effect of duration of light and dark on flowering. The effect of photoperiod or daily duration of light period on the growth and development of plant, especially flowering, is called photoperiodism. The role of photoperiodism in the control of flowering was demonstrated for the first time by W. W. Garner and H. A. Allard in the year 1920. They observed that Maryland mammoth variety of tobacco could be made to flower in summer by reducing the light hours with artificial darkening. It could be made to remain vegetative in winter by providing extra light. Later it was found that most plants would flower only if they were exposed to light for less or more than certain period, the critical photoperiod, each day. Subsequently, it was observed that is the light-dark cycle, dark period is crucial in initiating flowering and not the light period as thought the photoperiodic response. Another one, plasticity. Development of different kind of structure by a plant in response to environment or phases of life. The plant follow different pathway in response to environment or phases of life to form different kind of structure. This ability is called plasticity. The occurrence of more than two type of leaf on the same plant, that is called heterophyly, is an example of plasticity. Heterophyly is a regular feature in several plants like cotton, coriander, larkspur, mustard, pea, etc. In such a plant, the leaves on the juvenile plant are different from shape than those in mature plant called developmental heterophyly. In number of aquatic plants like ranunculus, limnophylla, the aerial and floating leaves are generally broad and usually unlobed, while submerged leaves are ribbon shaped, linear and are very much dissected is called environmental heterophyly. Another one, redifferentiation, refers to the loss of ability to divide by the products of de-differentiated cell. The product of de-differentiated cell or tissue which lost the ability to divide are called redifferentiated cell or tissue, and this event is called redifferentiation. Secondary xylem and secondary phloem form intervesicular vascular cambium. Secondary cortex and cork are the example of redifferentiated tissue, whereas the formation of callus from a parenchyma tissue in a culture medium is an example of de-differentiated tissue. From the differentiation of the growth in plant is open. The cell arising from the same meristem having different structure at a maturity. The final structure of a cell arising from the same meristem determined by the location of the cell. Another one, senescence, the period between reproductive maturity and death of a plant or a plant pot. Like other living organisms, plants also grow old and their life is ultimately terminated by death. As the young plant grow, it undergo aging and developing into mature plant in an ordinary fashion. The later part of the developmental process, which ultimately leads to the death, is called senescence. Senescence may be defined as the period between reproductive maturity and death of a plant or part of it. It is characterized by the collective, progressive and iterative developmental process which ultimately leads to complete loss of organization and function of the plant or parts of it. The study of plant senescence is called phytogerontology. Another one, synergistic, cooperative effect of two or more substances. The effect of one or more substances in such a way that both promote each other activity. Example, activity of auxin and gibberellin or cytokinin. Another one, vernalization. Accusation or acceleration of the ability to flower by a chilling treatment. Many plants, especially biennial, do not flower before they experience a low temperature. They grow vegetatively during the warm season, receive low temperature during winter, grow further, and they bear flower and fruit. The low temperature requirement for flowering was first noticed by Clippert in 1857. In winter varieties of cereal such as wheat, barley, oat, and rye, he observed that these varieties were sown in spring failed to flower the same year but grow vegetatively. Such winter variety, when sown in the autumn, they flowered in the spring of the same year. Later, Lysenko, a Russian scientist, discovered the cold requiring biennial plant have been made to flower in one growing season by providing low temperature treatment to water soaked seed or young plant. He called the effect of chilling treatment as vernalization. Vernalization is therefore a process of shortening the juvenile or vegetative phase and hastening flowering by a previous cold treatment. So the vernalization is the accusation or acceleration of the ability to flower by a chilling treatment. Thus, vernalization itself does not induce flowering, it prepares the plant to flower. Additional information. Negative growth. The term is sometimes used for senescence. Next one, etiolation, a phenomenon exhibited by green plants when grown in darkness. Etiolated seedlings are pale, elongated, and sticky with primary leaves, unexpanded and yellow. Another one, photomorphogenesis, the control exerted by light overgrowth, development and differentiation of a plant, independent of photosynthesis. The various photomorphogenic effects are regulated by a photoreversible pigment, phytochrome. Another one, phytochrome, a pigment protein complex which exists in two interconvertible forms, PR form and PFR form. The PR form, which is stable in dark, absorbs red light and is instantly changed to PFR form. The PFR form absorbs near 730 and is converted back to PR form. Another one, morphatins, synthetic growth inhibitor which inhibit the germination of seed and the growth of seedling, reduce longitudinal growth of seedling, reduce longitudinal growth of shoot and development of the leaf blade, reverse apical dominance, and promote the growth of lateral bud, causing a broom-like form of the plant. Another one, coconut milk. Liquid endosperma coconut show cytogynin-like activity. Another one, ascorbic acid, prevents over-ripening and browning of cut fruits due to its being antioxidant.